Okay, and our final presentation before afternoon tea is from Peter Pemberton. Peter has tertiary qualifications in information technology and ecology and environmental management. He's now a mature age student at the UNE, utilising both his computer and ecology skills to complete a PhD researching molecular ecology of outcrop endemic flora.
Good afternoon, everybody. My name is Peter Pemberton, PhD student UNE. Most students take a gap year. I took a gap half-life, but anyway, here I'm in. So I'm studying molecular ecology of endemic shrubs in isolated OCBIL populations. And here's a quote from Charles Darwin, who said, "Who can explain why one species range widely and is very numerous, and why another allied species have narrow range and is rare?" And it's those narrow range endemics that I'm that I'm interested in. So why old climatically buffered infertile landscapes? And I'll thank Steve for introducing OCBILs for me. I don't have to explain them. They have a high incidence of endemic and rare species. The ground outcrops can form terrestrial islands, and these terrestrial islands give you a novel study site. This particular picture here is one of my sites, Wombat Rock. We'll come back to that later. But the thing to note here is these outcrops can be made up of large boulders and rock sheets that are at different levels. So the questions I'm looking at, as well as the influence and geography of pollinators, genetic diversity in small isolated outcrop populations, and how do these populations maintain genetic diversity. So if we look at these terrestrial islands, especially with these shrubs, they have invertebrate pollinators, and these pollinators may not travel too far, so therefore you may not get the pollination gene flow between the outcrops. Also, these outcrops may be quite small, and therefore they've got very few individuals, so these populations are quite small, and small populations can suffer from inbreeding and also genetic drift, and both of those can deplete your genetic diversity within a population. Part of my study was a fine scale study on a Cyananthanthus and Flexus subspecies grandiflorus. There's five populations of this subspecies. It was thought to be extinct until some park rangers of Government National Park discovered it about 20 years ago. I think Paul in past life also discovered one of the populations, and 3 of the other populations have been discovered since. Wombat Rock is the one in the middle up top. The furthest distance between all these populations is about 1 kilometre. So what we did was a near complete genotype sampling of the Wombat Rock site. Of 164 individuals, we were able to get 162 data from 162 of those. We also used a advanced AMLID GPS system to take we plotted each site to centimetre precision, and that was important because this site's only probably about 60 metres by 40 metres, so we had to get that spatial data, we had to get that precision. We also took the height data of each plant as a proxy for age. First of all, we looked at we did a trial visitor survey of all the 4 subspecies of the Inflexus, and this was just to look at what was visiting the plants, proving that they were pollinators beyond the scope of this study. But the main thing is we're just seeing a lot of native insects, and we weren't seeing any birds or any vertebrates. So this was sort of lending itself that these invertebrate pollinators may not be transferring pollen between populations. So also part of the study I was doing population genetics, and of the other 4 populations I took 20 random samples, and I compared them to 20 samples of Wombat Rock using PCA analysis and also structure analysis, and what we're seeing is we can see those 5 populations are very much distinct, which gives you a little evidence that pollen or seed-derived gene flows between those 5 populations. When we did the structure analysis of all the 5 populations, also noticed that the population we're doing fine scale study on was also had a reasonable amount of structure to it. And what we did was, because we had the spatial data, each one of those columns in the structure map at the bottom represents one of the individuals, and we assigned the main structure to each individual in this slide. And we can basically see a lot of that structure was aligned to patches. And those patches are basically separated by rock sheets and large boulders, which sort of indicates that the boulders and the rock sheets were influencing pollinator movements. But it does also raise the question that these patches indicate family groups, which would probably further exacerbate genetic drift or inbreeding. So then we went on and did a heat map of kinship. And with the kinship, on the far right-hand sides of the scale, kinship of about 0.25 indicates parent-child relationships, also indicates siblings. And the idea was this, to understand whether those patches were family or ancestral. If they were family, they would have a lot of parent-offspring or sibling relationships. But as we can see from the heat maps, there's very few dark green squares in that heat map, which sort of indicates it's more ancestral. The other thing is, if we look at on the heat map probably in reverse order, we can see that the patches A, B, C, D, E and F, as you get further away from patch A, which is the red one on the top right, the patch F, as you go across the heat map, and it's probably not showing up too well there, but you're getting less and less kinship. And we did spatial autocorrelation, and the relationship between relatedness and distance basically was about 0 at about an average of about 12 metres. So we're not getting too much gene flow across a lot of distance. So you're not only not getting gene flow between populations, even though they're in close proximity, you're not getting much gene flow within a population. So we took this a bit further, and we did an identity by descent analysis. And with this identity by descent analysis, it allows you to divide up the degrees of relationship of each individual. Of the 162 individuals, gives you about 13,000 relationships. And the reason why we did the fine scale was to sort of understand the parent-offspring relationships and plot them and see how far the offspring were away from the parents. But when we finished this analysis, we saw that the parent offspring, which is the aqua square up in the top right-hand corner, had zero. There was 0 plots there. This is indicating that, of this population of 160 individuals, there was not one offspring amongst them. Yes, some of these plants are 2.5 metres tall, probably a 5 to 10 centimetre growth rate per year. So these plants are 25, 30, 40 years old, and none of the older plants have produced seed that are standing in that population or germinated in that population, which was quite a surprising find. The other surprising find on this graph too, it's a bit hard to see via the graph, it's down the bottom right hand corner, is the unrelated individuals. That's because there's 12,000 of those relationships are polled on top of each other, which means 92% of this population, even though there's 162 individuals in an area of 60 by 40 metres, 92% is unrelated. And that's a good thing for the population, because basically by random mating you're really going to be outbreeding. But what we thought now, what explains these two characteristics, and what we're proposing is that the seed has a dormancy, and that seed dormancy is obviously older than the oldest individual in the population, but we don't know how old it is. But with that seed dormancy, each individual, even though they're probably a lot of them a metre or less apart, that each individual is germinating from a seed that's differentiated by different generations. from the height data, we divided the population up into 3 groups, and those 3 groups were related to, we made them equal, and just, but there's a bit of a history on this this site. There was a fire in 2014, and as it turns out, that the larger group, the group between 1 metre and 2.5 metres, was probably, they survived that fire because they're amongst boulders, and that middle size group, 55 centimetres to 99 centimetres, were probably individuals that germinated straight after the fire. And the smallest group, the ones between 25 and 54 centimetres, have germinated since. And in the year 2000, there was 2020, there was a wet year, and there was a recruitment event observed then. So what we've seen is, what we looked at is another PCA analysis of these different age cohorts, and they're not strongly differentiated, so we're quite comfortable with that. But if in the graph on the right-hand side, we can see that each of these cohorts have got their own set of private alleles. And so what we're seeing here, that as each germination event is bringing back new alleles that have disappeared back into that population. So it's really reinvigorating the genetic diversity that's maybe lost through genetic drift over the decades. So in summarising, it's covered a fair bit there. So what may happen in a small disjunct population, you may lose genetic diversity through genetic drift. If they're disjunct populations, you don't get that gene flow to replace that genetic diversity because of barriers. And you also got a potential for inbreeding, which all reduces your genetic diversity. And genetic diversity is considered key to survival. It adds to your adaptive potential, and adaptive potential is what all living organisms use to adjust to changing environments. So from our observations, we saw there was zero parents in this population, even though some of the plants may be 20 to 40 years old, and also 92% of the population was unrelated. So the proposed explanation is that the seed bank dormancy is reinstating the alleles back into the population from multiple historical generations, hence the TARDIS, a bit of time travel for a gene flow. And yeah, the gene flow from the previous generations may be key to avoiding excessive gene drift. So survival of small isolated populations which seem to be living on the edge. For this particular population, you've got historical gene reservoir facilitated by seed dormancy, highly unrelated individuals germinating from different generations. Because of that, there's more opportunity for outbreeding within that population. That alleviates genetic drift effects, and it's basically a surrogate gene flow. And what we know in conservation biology, you like to have a whole heap of different populations that are in different areas, and you get that gene flow at the same time. This population sort of flipped that, and what you've got is a gene flow in the same area, but over time. And the seed dormancy is allowing the persistence in specialised habitats that maintain small populations like outcrops and OCBILs. So there's my acknowledgements, but this last slide shows a picture of a male carpenter bee. Originally they thought they'd just be outside Sydney and also on Kangaroo Island. However, they're flying around the Grant Belt too. And what the purpose of this slide is that without the pollinators, you don't get the seed bank that's preserving this genetic diversity in the seed bank. And when we look at fire regimes and controlled fires, we may sort of look and think, okay, we'll protect the outcrop so we don't burn the rare species, but these insects come from the forest matrix around that terrestrial island. So we need to consider that we don't destroy their habitat and destroy them, because without the insects, we don't get the pollination, don't get the seeds. Thank you.